Next Tuesday, no way.
Yeah, most that they don't taste. Yeah, if we're going to do it, we would prepare the tea. That's.
Towards neogenesis, which is going to be the production, and as it says here, how do cells in the body make glucose on their own when it is not available externally? So in terms of the study aims, you have to talk about the importance of glucose for daily life. Why is it so essential, and which organs are dependent on glucose? We'll talk about the non-carbohydrate sources that are used to make glucose through the gluconeogenesis pathway. Importantly, we will describe why gluconeogenesis is not just the reverse of glycolysis. This is an important distinction to make and can be a very easy point for students to get confused. We'll talk about glycolysis and gluconeogenesis, but importantly, we will talk about regulation. So we will talk about the individual steps, and we've spoken about those steps in glycolysis. We'll talk about gluconeogenesis, but remember in the previous lecture, I spoke to you about the regulation points, the point of regulatory control. These are very important in all of these pathways. And you'll see a common thread throughout. We'll talk about the issues seen in type 2 diabetes, and we'll also talk about the basic principles of the Cori cycle, which remember is the citric acid cycle or the tricarboxylic acid cycle. 3 different names for the same process. Space for your questions and the reference to your textbook. So if we start off with gluconeogenesis, as the name is saying, this is glucose, and neo is fresh, genesis, so glucose new creation is if we were to break those words down. So this is the synthesis of glucose from non-carbohydrate precursors, as we mentioned above. This is really important, particularly when we come to talk about the brain, because the brain is is one of the organs that uses glucose as its primary fuel. The other cells, so not an organ but the cells, are red blood cells that also use it as their own fuel. So very important for the brain, and obviously this is the reason that the organism is able to generate its own glucose, because we need to maintain the brain in that particular active state. So if we look at an average brain, how much do we consume per day? It is 120 grams of glucose per day, which is a reasonable amount, particularly when you think that the entire organism, so the whole body, only uses 160 grams. So remember, 120 goes to the brain, red blood cells are going to contribute to that 160 as well, to the 40 grams that are remaining, and then whatever else is being used throughout the body. So the vast majority is in those two contexts. But we only have 20 grams of glucose in body fluids, so we're nowhere near that. And then if we have it the storage form, we've got 190 grams of glycogen. So remember, this is the storage form of glucose in the body. So when you take those two together, we've got enough for about a day. And we'll talk about glycogen in the next lecture, lecture 12, where we talk about the details, how it's made, etc. So you can see we don't really have a lot that is available. So this glucose has to be maintained, and of course it's going to be important during periods of fasting, which is either on purpose, where we might fast for a period of time, even when we're asleep, for example, that period of time when we're not eating, or starvation, when food is limited and is not readily available. Now, in terms of where the glucose is made, the gluconeogenesis pathway, where does it occur? It is predominantly in the liver, although small amounts can also occur in the kidney. And the liver is always going to be the main focus, but don't lose sight of the kidney contributing. And so this glucose that is made by the liver and the kidney, and I'll put big lines under the liver because this is the bulk. What is the point of it? The point of it is to maintain glucose in the blood so that it can be used by the other organs. So remember, it's going to be the brain and the red blood cells predominantly. So that's really the key points of what we're trying to talk about. So let's move on to the synthesis, so the glucose synthesis. And what we see is that this gluconeogenesis pathway exists in most organisms. And so we talk about this from simple organisms such as bacteria all the way up to humans. When you see pathways that are shared across different types of organisms, it tells you that it is an important pathway. If you have a pathway which is unique to a single organism, then it's probably an adaptive pathway that is specific to that particular organism's needs because of its environment or its metabolism. But when you see it shared, then it's something that is fundamental. And we'll also see this when we come to talk about the tricarboxylic acid pathway, and we look at the process of generating ATP, commonality across different organisms. So this pathway starts off with those non-carbohydrate precursors, so pyruvate, and we spoke a lot about pyruvate in the previous lecture. And so pyruvate is converted into glucose. And so as we see here, non-carbohydrate precursors are first converted into pyruvate. So this is the intermediate molecule, because we then say here is then converted into glucose. And as we said here, pyruvate goes into glucose, so this is our intermediate molecule in this entire pathway. So what are the major non-carbohydrate precursors? And we've got three. We spoke about this one in the previous lecture, lactate. Remember, this was a byproduct of skeletal muscle, so actively working skeletal muscle. This was generated, and this, remember, is a byproduct. It can be used, it can be funneled in to make glucose. And part of this requires this enzyme, lactate, which tells you that this is the substrate, and this is the activity of the enzyme. Whenever you have a dehydrogenase, it removes hydrogen atoms. And typically, this will then lead to the introduction of a double bond. So this is what we will see, and certainly we'll see it in the diagram below. The other two are amino acids, and we know that we get lots of amino acids from our diet, but during periods of fasting or starvation, you can also break down muscle protein to generate individual amino acids. Now, typically when we look at amino acids, we think of protein synthesis, but we're now moving into the realm of broadening our knowledge. And so amino acids can be used, in this case, to make glucose. They can also be used to make nucleotides. So basis of DNA and RNA, and we will see this in much later lectures, but you can see that it's much broader. And then the last grouping is glycerol. And here we've got triacyl glycerol hydrolysis. A triacyl glycerol is a lipid or a fat. And we know that lipids and fats are very good stores of energy, and that at their heart, they have a glycerol backbone. And so glycerol is one of the key components of these molecules. Now, what we find though, is animals can't take this glycerol and convert it directly into glucose. So there has to be an intermediate step, which is what we see in the diagram below. So the glycerol is either metabolized by glycolysis, or it can be converted to glucose by gluconeogenesis. And so how do we determine which pathway? It's based on the needs of the cell, of the organism, and of the organ itself. So depending on the need, it can be shuttled across. And so we see the same thing up here with the pyruvate. Depending on the need of the organism, the pyruvate can go into multiple pathways and give rise to multiple end products. So this figure, entry point of glycerol into gluconeogenesis. This is going to be important because this is showing to you how glycerol can actually go into the pathway and in turn generate glucose. So we start off with our glycerol, and you can see that we've got 1, 2, 3 carbon backbone with hydroxyl groups attached. Typically what we will see is that when we want to increase activity, the cells will add a phosphate. So they take ATP, and using a kinase, they will transfer the phosphate. Now we know what the substrate is because the first part of the name is here, so it's glycerol. This is the substrate. And we know the activity of this enzyme, it's a kinase, it's going to transfer a phosphate. And so what we have here is a glycerol phosphate molecule. And again, the naming should be very obvious because it's just telling you what is the substrate and what have we done to it. So there is the phosphate. Now in this example, this phosphate is actually not going to be the point of reactivity, although this is something that we will see later. The next thing we need to do is that we need to remove these protons. So we have glycerol phosphate, which is now here. And again, this now becomes the substrate. And we have a dehydrogenase. And so we've got our coenzyme NAD+ being converted to NADH and a free proton. By removing this proton and this oxygen-carbon bond needs to be stabilized, and we generate this double bond. And so what we have here is dihydroxyacetone phosphate. So we can see these two groups, the group there and the phosphate. So that's all it's telling you. But this now allows this molecule to enter into the gluconeogenesis pathway. Okay, now I mentioned to you right at the beginning, gluconeogenesis is not the complete reversal of glycolysis. Make sure you highlight this, make sure this is really clear to you, because this is another area that sometimes students can get confused and think, well, if I go in one direction to make it, I can just go the reverse. And you should know that biochemical pathways, in some cases, can be directly reversed, but not in all cases. So if we now look at this from an energetic perspective or thermodynamics, we can see that the typical free energy in making pyruvate is minus 90 kilojoules per mole. Okay? So it's a reasonable amount. And you should remember from the previous lecture that there were 3 irreversible reactions. we had the first reaction, which was glucose being phosphorylated to make glucose 6-phosphate, and the net result, obviously, with the ADP, because we've got the transfer of the phosphate, this was minus 33 kilojoules. We had the fructose 6-phosphate, which was a key intermediate. It too was phosphorylated to give us our fructose 1,6-bisphosphate. And again, quite a lot of negative energy. And then lastly, there was the phosphoenol pyruvate, which through the addition of ADP led to the conversion of this molecule. So remove that phosphate to generate pyruvate. So you can see that here. But the phosphate that is being lost because it's being donated to the ADP, and we regenerated a molecule of ATP. So again, potential occurs there. Now, because these are, right again, irreversible, go this way, the back reaction does not occur. If we want to go backwards to make gluconeogenesis, these reactions have to be bypassed. Because remember, we cannot go backwards. These are 3 control points, so if we want to go backwards, we've got to find an alternate way, and this is what this bypassing is referring to. Okay, let me stop there because it shows you the chemistry of the reactions. And this is quite useful to see, okay, well, what is actually occurring? So I don't want to discount these, but remember, make sure you understand those key points that we're talking about here first before you start to get into this detail. So we started off, we had our pyruvate, and we've got this same type of reaction. So remember, we've got lactate and some amino acids can come in to give us pyruvate, and here it is. Carboxylase, so remember, carboxylase is going to have that carboxyl group, so here it is in the bicarbonate, and it's an ATP-driven process, gives rise to oxaloacetate. And some amino acids can feed into oxaloacetate, and again, oxaloacetate is very important when we come to make ATP through the TCA, citric acid, or Cori cycle pathway. Remember, same thing, 3 different names. We've got our phosphoenol pyruvate carboxykinase that gives us our phosphoenol pyruvate. We've got an enolase reaction which changes the configuration through hydrolysis. And then we had our mutase reaction, which again did a very similar process. We've got phosphoglycerate kinase, which again is taking the previous molecule, adding the phosphate, and we've got 1,3-bisphosphate, so it's telling you adding it twice. So these reactions occurring twice. And now we're moving into this intermediate glyceraldehyde 3-phosphate. We've got glycerol coming in, which enters with this substrate molecule. And this was the previous figure. This was the endpoint of the previous figure where we had the dihydroxyacetone phosphate. So what this is showing you above, remember we said that there were 3 non-carbohydrate precursors. We started off with amino acids, lactate, then amino acids, and then glycerol. And we've got that there. So this is showing you the entry point. Where do they come into the pathway? We're moving through. We've then got the aldolase. So we've got the fructose 1,6-bisphosphate. That can be converted to fructose 6-phosphate. That goes to glucose 6-phosphate. And here we've got glucose. So there should be some familiarity. There is recognition here of some of these steps that are the same as what we have seen previously. And so remember, we spoke previously about those steps where we've got commonality, and we also spoke about those areas that cannot be reversed. And that was the point of this whole lecture, or this part of the lecture. So let's get into some of that detail. So, conversion of pyruvate to oxaloacetate. So we're going through all of these steps one by one. So remember, this is a broad overview, but we'll also go through them stepwise. So it might be useful to annotate this diagram as you go for those important points, and also the key molecules that are important. And remember, there is overlap, so that will make things easier because you already know what's occurring with glycolysis. It'll make it easier to remember for gluconeogenesis, but the 3 points that are different are also going to be the points that we're going to focus on. Okay, so the very first step of gluconeogenesis is the carboxylation of pyruvate to form oxaloacetate. And this is achieved by the enzyme pyruvate carboxylase. Now, let's go through. There's a few points here that I want to make. First step. This is typically referred to as the rate limiting step because this will control the entire pathway. And I've shown this to you before just as an example using dominoes. That it's not until you push this very first domino that you go down the entire pathway. This is the first step. So if you control this first step, you then control the rate of activation, because if this only converts 1 substrate per second, which would be very slow, but if it did, then all of these are going to be limited based on this. Even if these can go faster, they're only going to be fed at this particular rate. The next step is to remember that we're starting off with pyruvate, and we said that before. Pyruvate makes its way ultimately to glucose. So this is what we're showing you here. The very first step is pyruvate. Remember that this is that non-carbohydrate carbohydrate intermediate. And how it's coming, we've spoken about lactate, amino acids, and glycerol. So one of those key intermediates is going to be oxaloacetate, and this is acted upon by the enzyme. And again, look at the enzyme name. We said, well, substrate is going to be pyruvate. It's going to get converted to oxaloacetate, which is the product. So the enzyme is pyruvate, and it's a carboxylation reaction, so it's a carboxylase. We've got the ASE to again signify that this is an enzyme. So we're just transferring a carboxyl or CO2 group onto it. This occurs within the mitochondria of the liver. So here is our pyruvate. We've got a carboxylation, so we know that CO2 is going to be there. We've got ATP, so this is an energy-dependent process, and a water reaction. So pyruvate carboxylase is shown in both directions, so it can be in either orientation in terms of the forward or the reverse reaction. So again, what you can see here is this group occurs, it's staying here. You've got the carboxyl group, and you've also got, which is coming from here, you've got this other carboxyl group there. So oxaloacetate is the key point. We've got ADP because we've used that phosphate for energy. Phosphate has been released, so you can see that this is not being attached. So it's not attached. If it's not attached, then it is just used for energy. Now, let me rewrite that because I did a really bad job of just writing the word. Okay, and we've released 2 protons. So this can also lead to acidification. Now, what are the key steps? It's a very simple reaction, but there are key molecules that are involved in this control. And the first one is biotin, and this is a vitamin. This is vitamin B7 derived, so again, it's one of those cofactors that we spent talking about in a previous lecture. This is the prosthetic group of pyruvate carboxylase. Now, prosthetic group is part of the enzyme, okay? But remember, as a cofactor, it's involved in carrying electrons or carrying part of the reaction and allowing it to occur. In the absence of the biotin, the pyruvate carboxylase is not going to be functional, so it's important. So biotin acts as an activated carrier of the carbon dioxide, and this is critical because remember, carbon dioxide is the additional group that's being added. This is an addition reaction because it's a carboxylation reaction. We're adding CO2. And this is a 3-stage process. We have bicarbonate, which is shown here. That's bicarbonate. Now, bicarbonate is part of the blood buffering system, maintains the blood pH around 7.4, but as a result, you have large amounts of bicarbonate available, so it's a readily available molecule. You've got ATP. ATP is going to phosphorylate the bicarbonate, and that's what we see here. We've got the phosphate that has been attached and the ADP that has been released. This leads to activation of bicarbonate. Now, when we see the word activation, what we're talking about here is the addition of the phosphate. Okay. Now, that activation is going to, again, facilitate the next stage of the reactivity, which is what I mentioned to you previously. We then have our biotin that is linked to the enzyme, what we've got up here. We've got our substrate, which was the activated bicarbonate, and now we've got CO2, which is bound, we've got biotin and the enzyme. And then we release the phosphate. And so what have we done here? All that we've done is that we have transferred the carbon dioxide to the biotin. Okay? So the bicarbonate is acting also as an intermediate because it's facilitating this transfer so that the CO2 now is bound to the enzyme. Remember, the CO2 is part of the reaction. For the reaction to proceed, it must be part of the enzyme, but for the CO2 to be captured, this biotin is required. And now, what are we going to do? If we just jump to the end, we're going to add the CO2 to the pyruvate because that's what was occurring up here. We've just now gone to this stage. All of the previous steps, we're now saying, well, how do we add that to this group? So we've got our carbon dioxide, which is bound to the biotin, which is associated with the enzyme. So this is now ready. It's a bit like the transition state. We've got to add energy. So there's another phosphate molecule that's going to come in, and we need our substrate, which is the pyruvate. These all come together, and what we now have is that the CO2 is going to be transferred onto the pyruvate. And so what we see is that the biotin and the enzyme, this is CO2 free. And what do I mean by CO2 free? Well, it's no longer binding a CO2 molecule. Why? Because it's gone to the pyruvate. It's just there. And when you add that CO2 to the pyruvate, you end up with your product, oxaloacetate. And so this is going through those steps that are important. Now, what we also see is that pyruvate carboxylase requires acetyl-CoA because this is an allosteric activator. So remember, allosteric is binding at a different site. That binding then stimulates the activity of that particular enzyme. So depending on the level of this, if this is present, and remember the acetyl-CoA is again one of those intermediates that signals to the cell about metabolism. If this is high, it means that we want to go down this pathway. It's able to stimulate this process. So pyruvate carboxylase is going to be important. The role of biotin in this assay is also going to be important. These three steps are just taking you stepwise through the process, but you can see it is the biotin and the enzyme that are the critical parts of this reaction. We've now generated oxaloacetate, and we then have to convert this to phosphoenol pyruvate. This now occurs within the cytoplasm. So remember, before we were talking about the mitochondria, we set that up here, right up here. We're now moving to the cytoplasm. So different locations, different assays. And why would that be? Part of the benefits of having different locations is the separation of substrates. If you have the first reaction in one location and then the product moves out and goes into a different location, there's no contamination, there's no competition, and it allows for it to occur in a different location. It might also be that the enzyme that does the reaction just happens to be in the cytoplasm as opposed to being in the mitochondria. So, as we said, oxaloacetate must be transported from the mitochondria to generate phosphoenol pyruvate because it's in the cytoplasm. So that tells you why the transportation is important. And the very first step is a reduction reaction. So this reduction leads to malate. And so we have malate dehydrogenase. So remember, this is an example of a redox reaction. The reduced malate is then transported to the cytoplasm. So remember, this is occurring within the mitochondria first because it may not be able to exit on its own. So there's this conversion. It then gets oxidized back to oxaloacetate. And this occurs through an NAD plus linked malate dehydrogenase. So what we have is oxaloacetate going to malate. That can now exit, and then we go back to oxaloacetate. But remember, this is in the mitochondria. This is now in the cytoplasm. So what this is telling you, why does the cell do this? Oxaloacetate that is within the mitochondria cannot be transported out on its own. It has to be converted to a different molecule, transported out, and then converted back again. You might think that this is a wasteful process, and to some degree it is, but it also helps to facilitate the trapping of this molecule in one location. And we will see that oxaloacetate is really important when we come to talk about energy, and all of that is occurring within the mitochondria. Now, because we've got NAD+, and we've got a dehydrogenase which is going to strip the proton, this leads to the generation of NADH. And this is a really important process because this is then used in later steps. That reductive potential of the NADH is important. So, all that this is showing you is the reactions to allow oxaloacetate to exit the inner mitochondria and to move to the cytoplasm. And this is just so gluconeogenesis can continue. And as it says here, note the generation of NADH. So highlight that because this is important because it's used in later steps. Where does it come from? This is where it comes from. So if I zoom in to this diagram, you can see here's the mitochondria, or mitochondrial, because it's a single one. You know that there's a double membrane. You've got this inner level or inner imagination of the mitochondria, which increases the surface area. So this is the cytoplasm. This is the inner matrix of the mitochondria. Pyruvate undergoes that carboxylation reaction to give you oxaloacetate. Oxaloacetate can't go out. So what happens? It's converted to malate. And here we've got a transporter protein. This is just a protein that is able to shuttle molecules out of the mitochondria into the cytoplasm. And so it shows you that it's being transported. The malate is then converted, so again NAD plus goes back to NADH, to oxaloacetate, which is the molecule that we were after. Okay. Now, what happens to the oxaloacetate? It is simultaneously, which means that two reactions can occur, but it doesn't really matter in terms of the process. It is first of all decarboxylated. So remember when we had a dehydrogenase, this refers to removal. So carboxylation, we're going to remove CO2, and it is then phosphorylated by this enzyme. Phosphoenol pyruvate, so we know what the molecule is, carboxy, and it's a kinase. And so remember, we said phosphorylation, so hence the kinase. We've got the different groupings that are also referring to the substrate and the end result. Why is this important? Decarboxylation often drives reactions that are highly endergonic. So we've got the citric acid cycle, we've got the pentose 6-phosphate pathway, which is going to be coming up in a future lecture, which is all about dealing with oxidative stress and fatty acid synthesis. So what is the net reaction? We've got pyruvate, we've got ATP. Notice that we now have GTP, a different molecule coming in, but it's doing essentially the same thing. It is a phosphate donor. The only difference is the letter, so the base is different. So this is adenine, this is guanine. We've got hydrolysis reaction, so we've got phosphoryl pyruvate. These are both going to expend some phosphate. There's a free phosphate molecule and some protons. So this is the net reaction of what we have just seen. It is important to know those intervening steps because it's telling you what is occurring. Okay, one last bit before we stop for now. What is the whole point of this? The whole point of this is that these reactions bypass the irreversible reaction which is catalyzed by pyruvate kinase. The whole point of this is to be able to reverse the reaction, but we can't do it using the traditional approach, so the cell has to use alternate pathways. So we're not using pyruvate kinase. And so what we can see here, oxaloacetate through the phosphoenol pyruvate carboxykinase, PepsiK, gives you phosphoenol pyruvate, which is that initial overcoming step. So here's our GTP, goes to GDP, and we've decarboxylated, we've lost CO2. And again, one of those points to highlight, note GTP involvement as opposed to ATP involvement. Okay, let me stop there. When we come back, we'll go to the next reaction that allows us to go backwards. Okay, welcome back. So we just finished talking about phosphoenol pyruvate. So we started with oxaloacetate, moving to phosphoenol pyruvate as the first step, as a bypass mechanism. The next one is fructose 1,6-bisphosphate, and we want to convert it to fructose 6-phosphate. So phosphoenol pyruvate, which is the previous substrate that we just generated, this is next metabolized by enzymes of glycolysis, but it's in the reverse direction. So here, the reverse direction can occur. So that facilitates it. So remember, we don't have to reverse, we can't reverse every reaction, but we just need to focus on those ones that we can't, and we have a different mechanism. The ones where we can, then it's just going to be the reverse of what we already know. So this occurs when gluconeogenesis is favored. What does that mean? It's when there is a requirement within the cell, that there is a requirement for glucose production as opposed to glucose breakdown, okay? And this keeps going until the next irreversible step. So it goes as far as it can until we get to the next roadblock. And that is going to be the conversion of fructose 1,6-bisphosphatase, or phosphate, to fructose 6-phosphate. Okay, so not ACE, it's phosphate. That's the enzyme, and that's what we're talking about here. So this is catalyzed again by an allosteric enzyme, which is fructose 1,6-bisphosphatase. Now, we know that a kinase adds phosphate. What reverses that? It is a phosphatase, and that removes phosphates. So you can see here, fructose 1,6-bisphosphatase, we're removing a phosphate. So here is the fructose 1,6-bisphosphate, one phosphate, the second phosphate. This is shown in a reddish color, because that is the group that is going to be lost. We want to remove that phosphate, it's a hydrolysis reaction. So here's water, and here is our phosphatase. We remove that group, and the water leads to the addition of that hydroxyl group there, with a proton in this case. And here's our free phosphate. This is then converted to fructose 6-phosphate. Now, what about the generation of the free glucose? So fructose 6-phosphate is readily converted into glucose 6-phosphate. Remember, this is one of those steps that we saw before. Now, in most tissues, what we see is that this reaction is going to stop here. Glucose 6-phosphate is converted into glycogen, and this is a form of storage, an efficient form of storage of glucose, but it is limited because we can only really store a certain amount because it doesn't give us that much energy in comparison to, say, fat. So the final step, again, is now going to occur principally in the liver. And again, this is always going to be the main focus of what we're talking about. Glucose 6-phosphate is transported into the inner portion is what the lumen refers to, the inner portion of the endoplasmic reticulum. Notice that we're going in different organs, we're going in different locations within the cell. Again, this is part of that segregation and that separation of substrate and also the location of an enzyme within the cell. It too is hydrolyzed to glucose by glucose 6-phosphatase. So remember, we're starting off with fructose, 6-phosphate. We need to remove that phosphate, so we need another phosphatase. And that glucose then gets shuttled back to the cytoplasm. So you can see this interconversion and movement. So glucose 6-phosphate, phosphatase, another hydrolysis reaction to give glucose. This is the same as above in terms of the reaction. And what do I mean by above? By this reaction. We had phosphate, we had a hydrolysis reaction, and we had the loss of a phosphate molecule. It's the same reaction, just occurring on a different substrate. Here it's glucose 6-phosphate to give us glucose. You can see what's occurring here. We've lost that phosphate to here. And really could put an arrow to show the reaction that's occurring. Now, this is showing you in this diagram here, glucose 6-phosphate is transported into the endoplasmic reticulum. There is a specific transporter, transporter 1. It's converted into glucose and then transported back into the cytoplasm. There are two different transporters that allow this to occur. So you can see here on the outside, so the cytoplasm, glucose 6-phosphate comes in. Here it is. We've got plenty of water because remember, water is a ubiquitous molecule. We've got our phosphatase, we lose the phosphate and we've now got glucose. The glucose is not going to be useful to us inside the lumen, so it has to come out. And so we've got two different transporters that allow it to come back into the cytoplasmic side, where other substrates, other enzymes are going to be present. Okay, now what is the energy cost associated with gluconeogenesis? We already know that this is going to be an energy-intensive process because we've seen the involvement of multiple stages of phosphorylation. So gluconeogenesis is energetically unfavorable unless it's actually linked or coupled to favorable reactions. And we will see this a lot in biochemistry. You might have a reaction that is like this. A plus B gives rise to a product. This is favorable. And then you've got another reaction which is not favored. But if you then couple or you link these two together, so there's some interaction, actually if you make that B plus E gives rise to F, you link these together, it drives the net reaction forward. And that's something that we've seen. In terms of energy, if you count it up, 6 nucleoside triphosphate molecules, and remember it's a combination mostly of ATP, but we did see GTP, are hydrolyzed. They are consumed. They have expended their phosphate. And so they need to be consumed so that we can synthesize glucose from that original pyruvate molecule. But only 2 molecules of ATP are generated in glycolysis. So the net result is -4 phosphates. We have had to expend 4 phosphate molecules. So therefore, if gluconeogenesis was simply the reverse of glycolysis, it would not occur. So if I was to ask you, why is gluconeogenesis not the reverse of glycolysis, you would have 2 reasons. One would be the inability to reverse some reactions, and the second is obviously the amount of energy that is required. And that's part of the unfavorable nature of what is occurring. So, nucleoside triphosphate coupled reactions, what does this mean? ATP or GTP involved reactions drive an energetically unfavorable reaction. So energetically means that we must input energy. And where does that energy come from? It comes from those two, from the expenditure of energy from ATP mostly, but also some GTP that is occurring. And you know this should make sense. If you want to do something that doesn't require energy, you've got a ball at the top of a hill and you want to let it go, just gravity does it, it happens easily. If you want to then push that ball up the hill, you're going to expend energy because you're going against gravity. It's not a favorable reaction. It's the same logic here within the cell. Some of those steps are not favorable, so energy has to be put in. Now, even though this is a negative input, that we're losing energy, remember that through metabolism, we're going to be generating lots and lots of ATP molecules, far, far in excess for this. Otherwise, this would not be sustainable. So there's a loss here, but the net gain is going to be far, far higher later on. So it's worth expending some time and energy in this situation. Okay, let me stop there because as you can see, far in excess for this. Otherwise, this would not be sustainable. So there's a loss here, but the net gain is going to be far, far higher later on. So it's worth expending. Okay, we'll come back. So, regulation of gluconeogenesis and glycolysis. We can talk about these together because we have spoken about them separately. We know where there are points of overlap, and we now know where there are points of difference. So what you see is that there is a coordination between each pathway. And remember, this is production, and if we use the word consumption for the other pathway, but it's the opposite effect, okay? So if we have these two opposing pathways, what we need to do is that we need to think that when one is active, the other one needs to be inactive. Otherwise, we're going to be sort of caught in a bit of a loop, and it's going to be quite wasteful to the cell. And as we saw above, that can be energetically unfavorable and certainly comes at an energetic cost to the cell. Now, both pathways are going to be exogenic under cellular conditions. So that means that it's possible for both pathways to be active, which would be a problem for the host and for the cell. But what we know is that there are going to be activities of distinctive enzymes in each pathway. There are key enzymes that control each pathway. And as a result, this allows for both pathways not to be active at the same time. If you have the rate limiting enzyme, if you have a point of control that is at the beginning of each pathway, you can in turn either turn it on or off. And remember, if one is on, the other should be off, and reciprocally, if one is off, the other should be on. This is an easy way to do that. You only need to control that key enzyme, and you control the entire pathway. So please highlight these 2 points. These are really important. The rate of glycolysis is determined by the concentration of glucose. That should make sense. This is the substrate. So if this is limiting, you don't want to break it down. If you've got an excess of glucose, then you're going to want to break it down. Similarly, the rate of gluconeogenesis is determined by the concentration of lactate and other precursors. So remember those non-carbohydrate precursors that ultimately go into pyruvate, and that pyruvate gives rise to the glucose. So look at this from really the top-down approach. We're starting at what feeds into the pathway. Whatever feeds into the pathway is going to be the molecule of control. So when glucose is abundant, glycolysis is going to predominate. There's no point making glucose and activating gluconeogenesis when there is ample amounts. It would be wasteful, it would be silly to do so. When glucose is low, you will see that gluconeogenesis will occur, and so vice versa. If you don't have a lot of glucose, the last thing you want to be doing is to be breaking it down. You'd rather be synthesizing additional glucose so that there is a buffer, there's an ability to store, and that the brain and the red blood cells are not going to be deficient in this vital energy source. Key regulatory sites of gluconeogenesis in the liver, because again, we're talking about the liver, we're not forgetting the kidney, but really this is where 99% of the activity is going to take place, is the interconversion of fructose 6-phosphate to fructose 1,6-bisphosphate, and phosphoenolpyruvate and pyruvate. These are the key molecules, and we'll look at 2 different scenarios depending on which is occurring and also the energy requirements. So again, highlight these important molecules. Notice that we're talking about the same molecules in either glycolysis or gluconeogenesis. We're talking about the importance of these molecules in these pathways over and over again. This will be my focus moving on, okay? And this should also be your focus when it comes to, say, an assessment, because
the key regulatory sites of homogenesis is
this is really the key points that we're trying to understand. Okay, so let's have one example. Scenario number 1, energy is required. If AMP levels are high, remember AMP, so we've got ATP gives rise to this, so energy has been expended. If we've got a lot of AMP, we're driving this reaction forward. AMP will stimulate phosphofructokinase, or PFK. And by doing this, by stimulating this one, it also has the opposing effect of inhibiting fructose 1,6-bisphosphate. So the net result is glycolysis is activated because we need to break it down, and gluconeogenesis is turned off. Because remember, we've got a lot of this, which means that we're running out of this. So we need to replenish. So that should hopefully that makes clear sense. And this reciprocal nature, so one molecule can stimulate one key enzyme and inhibit the other, is again something that we will see over and over again, not just in this lecture, but in future lectures. Now the opposing scenario is where we've got excess energy. There's a surplus to our requirements. So we've got high levels of ATP and citrate, and this is now a new term or a new molecule. But what you need to know is that citrate is one of these molecules within the tricarboxylic acid cycle. What this is telling you is that biosynthetic intermediates are abundant. We have lots of precursors available. Okay? The organism is happy, there's lots of energy. Both ATP and citrate, which are these precursors, these will inhibit phosphofructokinase. So we had an example up here of stimulating, now we've got the example of inhibiting. Now remember, in the previous example, we said that AMP stimulated PFK to make ATP. Not surprisingly then, if ATP levels are high, it's going to be a negative effector on PFK. Because if this stimulated, well then the opposing molecule must do the opposing effect. And we see the same thing with citrate. But I've focused on ATP because we've got this lovely example above, and this now shows you the opposing effect. Both of these will activate fructose 1,6-bisphosphatase, which remember, took off the phosphate. And that basically means that glycolysis is now turned off because we've turned it off up here, but remember the phosphatase turns on gluconeogenesis. So we're going to stimulate glucose and allow the process to move forward. So, regulatory controls for glycolysis and gluconeogenesis. Notice we've got glycolysis on one side, gluconeogenesis on the other side. We've got the pathway going from top down, depending on which way we want to go. So glucose, fructose 6-phosphate, fructose 1,6-bisphosphate, phosphoenolpyruvate to pyruvate, which is the glycolytic pathway, and the gluconeogenesis was the opposite direction. So pyruvate was converted to phosphoenolpyruvate, 1,6-bisphosphate, fructose 6-phosphate, and we're making glucose. So that's how we know we're going in the right direction. Now there are lots of different examples on this side. I think it's important for you to understand the ones that are stimulators are shown as positive and the inhibitors are shown as a negative. And again, make sure you understand these two here are going to activate PFK, which we saw previously. We saw that ATP and citrate would inhibit PFK, and that in turn dictates which direction we're going. And we have a similar thing further down, but we've got different molecules that are playing a role. And notice here, amino acids coming in, okay, alanine. And again, we've got same molecules playing a role on the opposing side. And again, another key molecule, acetyl-CoA, which is very much important for the citric acid cycle, tricarboxylic acid cycle, and the chlorine cycle. We'll see that later. So this shows you the key molecules. You would know these now. And remember, some of these steps are not shown because they're not the points of control, and that's okay. But you understand that you can stimulate and inhibit different pathways. Okay, so gluconeogenesis and glycolysis regulation in the liver. Why do we need to do this? The rate of glycolysis and gluconeogenesis are adjusted to do one thing: maintain blood glucose concentrations, first of all. The second point is that they have to be maintained within tight limits. If you have too much glucose, over time, that leads to pathology. Too little, that can lead to death. Because we know that glucose is going to be very, very important. Because remember, it's the only carbon source or the only energy source for the brain and red blood cells. If we ignore the red blood cells, it's obviously going to be very important for the brain. So if you ever meet someone that is a diabetic, you know that they don't want to be too low because it can lead to seizures. And for prolonged periods of time, it can lead to death. But at the same time, if they have excessively high levels, this can lead to different types of pathology. And this is where they can have issues with blood flow to the extremities, and this can then lead to amputation. when you've got too much. So the tight limits are very, very important. And remember, another way of talking about tight limits is that we're trying to maintain the homeostatic level and maintain homeostasis within the organism. So you should remember, fructose 2,6-bisphosphate activates phosphofructokinase, and it's the main regulatory enzyme in glycolysis. And as we said before, this inhibits fructose 1,6-bisphosphatase, which is involved in gluconeogenesis. We've got one pathway controlling the other. So let's look at an example. We've got blood glucose levels that are decreasing because we've fasted overnight as a result of sleep, or we've purposely not eaten. What happens is that fructose 2,6-phosphate is dephosphorylated and gets converted to fructose 6-phosphate. Phosphofructokinase is now less active, and we've got gluconeogenesis predominating. So what are we doing? We are making glucose because glucose levels are low. Because we fasted overnight, we've been asleep, or we haven't eaten for a prolonged period of time because food is not available. So it shows you that the body is going to respond, and it shows you again those control points. And these control points and mechanism of control is what we saw above. So, how is fructose 2,6-bisphosphate actually controlled? This is interesting because there are actually two enzymes that control the molecule. One which phosphorylates it, and the other one which dephosphorylates it. So again, in simple terms, you know this now, that this is the kinase, and this is going to be the phosphatase. This can be the on switch or the off switch, and it can be vice versa depending on how it is used. But that's a simple way to think about it. Typically, you add a kinase to turn something on, and you remove the phosphate to turn it off. We do see alternate situations, but that's just a good rule of thumb. But what we find is that both of these activities are present within the same protein. So you might be thinking, well, hang on, we've seen this before, this is nothing new, but we see this from two proteins, two enzymes. What we're talking about here is actually within the same protein. The same protein has a kinase domain and it has a phosphatase domain, which is quite unique. And this is what we see here. We have an alpha helix. Here, we've got the hooks and turns. We have some beta-pleated sheets, not many. Here, in this domain, there's a little bit over here as well, on this side here. So what we can see here, this is the amino acid sequence, so position 1 all the way to 470. You've got the regulatory domain, which is at the beginning, so 1 to 31 probably. You've then got the kinase domain, which is this part of the protein, and then you have the phosphatase domain, which is this part of the protein. So really quite interesting. The kinase and the phosphatase domain are within the same protein. So this protein is set up for turning on and turning off the reaction, and that is its principal function. Okay, let's stop there because I want to move on to gluconeogenesis with diabetes, and we're going to go into a slightly different context, and just to make this lecture live. Okay, welcome back. So we just finished by talking about the kinase and phosphatase domains within the enzyme, and now we want to talk about a clinical insight. So this is giving you a bit more of a day-to-day perspective. And we're going to talk about type 2 diabetes. So insulin fails to inhibit gluconeogenesis in type 2 diabetes. Now, insulin is a hormone, and you should know that it is made in the pancreas by the islet cells in the beta region, islets of Langerhans. I'll just remove that bit, don't worry about that for now, in the islands. So this signals the presence of fuels, and remember when we're talking about fuel, we're talking about glucose in the blood. It is the marker, it is the hormone that is responsive to the level of glucose in the blood. So when glucose is present, remember that gluconeogenesis is not required. It is in excess. So after a meal, for example, you've had some lollies. Easter is coming up, okay? I know I'm going to have probably too much chocolate than I should, but I'm going to have a sugar hit. That sugar, which is going to be predominantly glucose, is going to raise my blood glucose levels. So making additional glucose would not make sense. So my cells won't be doing that. They'll be going down the alternate pathway, which is glycolysis. So insulin turns on the genes involved in glycolysis, as I've just said to you, and it turns off the genes involved in gluconeogenesis. And this is the normal state. Now notice here that we're talking about genes versus proteins. All above we have spoken about proteins, because remember these are the active state. These are the ones that are actually performing the function. But the gene gives rise to the protein. So if we really want to turn off a pathway, we turn off the gene. Ultimately, the proteins, after they go through their half-life, because remember, a protein can only exist for a certain period of time, if this is turned off, the gene is turned off, the level of protein is going to decline. And that's an easy way to control and make sure that that pathway is in fact turned off. Don't have functional enzyme, you can't have a functional pathway. But in type 2 diabetes, even though insulin is present, phosphoenol pyruvate carboxykinase, so PepsiK, remains, and it still stimulates gluconeogenesis. So remember, this is now a disease state. The effect of the insulin is being ignored. We should be turning things off, but the enzyme is persisting. So what is the end result of this? We have a higher output of circulating glucose. So it's coming from the diet, but also through its own production. And remember, if I scroll up here, remember I said, too much glucose gives rise to pathology. And what are we doing now? We are making too much glucose because A, it's in the diet, and also the cell is actually making its own glucose through gluconeogenesis. So what this does, excess glucose actually changes the osmolarity of the blood, and this leads to polydipsia. Poly just means many, and this is referring to excessive thirst. So you notice people that are diabetic are going to be always thirsty because they're, and then remember, now, metabolism in context. So lactate, we know, is produced by active muscle cells and red blood cells as well. And remember that this can also be a source of energy for other organs. The body typically does not produce products that are wasteful. So when you have a product such as lactate, it will be used by some other pathway. Any metabolite that is made that has some opportunity will feed into another pathway. The body is very efficient, doesn't want to generate things that are wasteful. So, in active muscle, and that's important, actively exercising muscle, pyruvate levels will exceed the rate at which the tricarboxylic acid cycle can metabolize it. And as a result, lactate dehydrogenase will convert excess pyruvate to lactate. But we've got a problem. Lactate is a dead end. It's got to be converted back to pyruvate for metabolism. So yes, it's a dead end, but there are steps that can take place for metabolism to occur. And so in the muscle, because this is the location that we're talking about, formation of lactate occurs in the absence of oxygen. It allows it to make ATP, and so what this does is it actually shifts the burden to metabolize lactate to other organs, and in this case it's going to be the liver. That lactate moves to the liver and is then metabolized so that we've got an alternate source. And the liver restores the lactate back to glucose. Remember, we've got to go back to that process. And that in turn can be used by the muscle cells, and this is the Cori cycle. And just thinking, I think I mentioned it incorrectly before that I was linking this to the citric acid cycle and TCA cycle. What I was thinking about was actually the Krebs cycle. So please ignore when I talk about Cori cycle, linking that in. These are different pathways. That was my mistake. So Cori cycle is the ability to take the lactate, convert it back to glucose so that it can then be used by the muscle. So that's why it's a cycle, because we're going through that round process. Okay, so the Cori cycle, as it says, lactate in muscle, because this is where it's being